Hello all, in this video session we'll discuss in situ hybridization. I'm Moon Chatterjee, I'm postdoc at Yale in the lab of Dr. Thomas Pollard. So in situ hybridization is also called as ISH. We'll first look at the textbook definition of ISH. So it's a method to probe the presence or absence of DNA or RNA in specific cells. The words to be emphasized are probe. So we are identifying molecules of DNA or RNA. We'll discuss how and why we need to do it and why do we need to do in specific cells at all. To be able to understand this method, I will divide this video into 4 components. To start with, we'll first think about what it means generally. Followed by, we will think about some of the concepts, which are mostly simple and you are familiar to. This will be followed by discussing applications of in situ hybridization. And finally, we will look at the methods to detect signal for in situ hybridization. Now, if we think about the word in situ hybridization, it decomposes into 2 components. The first component is in situ, which means on site, and the second component is hybridization, which means mixture of 2 kinds, right? So on site in this context means or can be tissue, it can be embryo, it can be infected cells or cancer cells. Now let's think about hybridization. So remember, we are trying to identify nucleic acid molecules, so DNA or RNA. We do so with the aid of other molecules of DNA or RNA. To facilitate identification, we tag these, and these tagging can be by various methods. It can be fluorescent, it can be with antibodies. Here, we mostly will discuss fluorescence. Let's think about some concepts. The first concept I want you to think about is complementarity. So, nucleic acids are made up of bases, which when can find complementary bases, can hybridize. So ISH exploits these properties of nucleic acids. It uses these hybrids, which are double-stranded strands, to generate signal and be able to identify nucleic acids. Next, let's look at the second concept. I want you to think about cellular identity. What I mean by this is what is special about a RBC or what is special about a hepatocyte. So the identity of cells is mostly imparted by its proteins. So each cell type expresses a subset of proteins. Now let's think about central dogma. So we know DNA in the nucleus transcribes into multiple copies of RNA, which can be in the nucleus or cell, which translate into multiple copies of proteins. Now let's think what all these concepts, how these translate into in situ hybridization. So looking at an embryo, imagine a gene A expressing in only one stage. So the RNA will be abundant in one cell. So RNA will be abundant in that particular cell. We add fluorescent probes to all over the embryo and then we wash it. So any RNA fluorescent probe which did not hybridize will be washed off, and we will receive signal only from the cell which has a hybridized molecule. We can detect that signal from that particular cell, implying that this gene A is being expressed only in that particular cell. Now, let's think about a cancerous cell. Now, this cancerous cell is cancerous because of gene duplication. We add fluorescent probes. We look at the control cell. We see basal level of signal. While when we look at the cancerous cell, we see enhanced signal, implying gene duplication was the reason. Now let's think about detection. So remember our probes. Our probes are molecules of nucleic acid. These are tagged. In case of fluorescence detection, the tagging is done by fluorescent molecule, which is a fluorophore. What we do here is we shine light, UV light from the electromagnetic signal. What it does is the fluorophore absorbs the light, it gets excited, electrons jump from the excited state, ground state, and move to the excited state. This is because of the absorption of light. This is followed by the electron returning back to the ground state, emitting light, and exhibiting a phenomena called fluorescence. This can be recorded on the detectors of your camera, which then can be seen as fluorescence signal on your computer. Now, signals, images of fluorescent signals are very pretty and beautiful. I have to admit, this is one of the reasons I love doing my work. Now, I hope in this video I have explained you the method briefly though of ISH, and I have shown you some of its applications. Stay tuned for more science videos. Thank you.